Thank you once again for the invitation to join this meeting. So as I mentioned, I'm from Lithuania, from the neighboring country, and my team is working in the microbiome field, and recently we have started to work with this very novel and exciting area of circulating microbiome. And today I will be presenting one of the works which my group has recently published. So starting from the very beginning, as if we heard from the previous talk, we have already been in this human microbiome field for quite a while, but I think that we're just at the starting point, but due to all this knowledge and all these exciting projects, we have gained so much knowledge during these 20 years that we can now say that we know a little bit about the functions of a bacteria, we know what we have and what might they be doing in there, and all this knowledge and all the technological breakthrough help us to enter this other field of a low biomass microbiome. So human blood as itself has been always thought to be sterile and basically consisted of blood cells and platelet and plasma, and bacteria in the blood had been always thought to be the sign of an infection. But going back in the timeline, we already see that the first signatures that the healthy human blood has bacteria, microbiome in it, have already been recorded in the 1970s, when the first paper showing up that there is the bacterial metabolic activity in the blood. The molecular biology-based knowledge using qPCR and fluorescent probes has been shown in the early 2000s. And the breakthrough due to the NGS has made all this knowledge which we have today, and has confirmed that there might be bacteria in the blood and that they might be doing something in there. So the next question, so how do they enter there? So is the blood the true biological niche for them? Are they residents in there, or are they just transient bacteria? So several of the hypotheses exist so that we can enter our circulation through the other organs like skin, oral microbiome, or gut microbiome, through the leaky gut, through the infections, through the injuries or medical procedures. And the other hypothesis is vertical transfer through the mother-to-child transfer. So based on the current knowledge, we already know a bit what our healthy blood microbiome could look like, and it does not resemble that of the other known microbial niches. So if we agree, or if we at some point have a knowledge that the blood microbiome is an ecological niche, we will have then a lot of other questions which would need to be addressed, like does the environment impact them? What other factors contribute to the composition of the blood microbiome? So the main criticism and the main challenges of these low biomass microbiomes, which I wanted to stress here as well, is the contamination possibilities, which stem from very first steps, from sample collection, from working with biological material, and ending up to the very last steps of the sequencing. But scientific communities know about these risks, and they are extensively working in the field to provide recommendations, to provide databases, to collect the data, and to develop the tools that would help to identify contaminant bacteria, and to ensure that the data we have could be interpretable. So coming back to my topic, so today I wanted to present the liver inflammation caused circulating microbiome changes. So we know that liver is one of the organs which is closest to the intestine, and there has already been shown that the gut microbiome of the liver patients is different compared to the controls, that there are changes which show that there is a direct interaction between these two organs. And it's extensively discussed in the literature, this so-called gut-liver axis. And we know that this interaction between the two organs takes place through the biliary tract, through the various circulating systems. And so it makes a very good candidate disease to study the circulating microbiome changes. So there are already several publications published in the field, which of course had different aims within their projects. Some of them looked at different compartments of the blood throughout a person's body and wanted to define the circulating microbiome. Others had a look into the virus-induced blood microbiome changes. However, they uniformly say that yes, there is an increase in circulating bacterial DNA in the blood. The diversity of the blood microbiome is decreased, and there are phylogenetic differences comparing control individuals and patients. However, these phylogenetic differences are not very consistent and vary between the studies, but it's very difficult to compare the studies due to the starting biological material, because some of the studies analyzed the whole blood, others took just the plasma samples, and had different analytical tools, different extraction protocols. So it's difficult to say which of them is the true picture of changes. So coming now to the paper I wanted to present today is the paper from our group where we analyzed a unique cohort of cirrhotic patients who had to undergo the hepatic vein pressure gradient measurements. And there we had the possibility to take the blood samples from the hepatic vein as well as from the peripheral vein. So we had the possibility to assess not only the microbiome in the peripheral circulation, but also to look, maybe there are already differences in the vein, which is very directly inside the liver. We also assessed inflammatory cytokines as well as leaky gut, so-called biomarkers, to see whether there are associations with the circulating microbiome. So the first thing we had a look, so here at the very bottom, if you see, so it's the hepatic vein and peripheral vein, so we did not see any differences between these two compartments. So it was kind of a go that we can compare when the control individuals and the cirrhotic patient individuals where we collected blood just from the peripherum. And here we see at the C figure, here, I don't know whether you see my laser, so that these two groups are totally different. So their microbiome is not the same. And these differences can be seen already on the very high level of phylogenetic analysis, where we see that cirrhotic patients have more of a Firmicutes phyla. And when we got to the genus level, we already identified very known factors for the liver diseases like Provatella and Escherichia shigella. So we were there in the circulation. As a next, we wanted to have a look whether there is an association or correlation between those inflammatory and leaky gut biomarkers with the circulating microbiome. So as expected, we saw that there are differences between the patients and controls when we looked at the biomarkers themselves. But when we made analysis with a circulating microbiome, only the inflammatory biomarkers, so EL8 cytokine level, correlated moderately with some of the bacteria taxa. And again, we saw the highest correlation with the Prevotella and Escherichia shigella, which kind of made sense, and because they were known factors for liver disease and pro-inflammatory bacteria. And as a next step, we wanted to have a look at those extensively collected clinical data, so this portal hypertension measurements, and to have a look, maybe they could be also used for distinguishing patients within the clinical setting. So we compared patients who had already clinically significantly increased portal hypertension with the patients who did not have this increase, and we saw that, again, certain bacteria are increased in the patients with clinically significant hypertension. And again, we see Escherichia shigella, Prevotella, Bacteroidetes in there. However, when we made the ROC analysis, we did not see any significant difference. So we kind of made a final conclusion that these bacteria cannot be used for differentiation on the clinical setting. However, our study results indicated that through the increased leaky gut, Prevotella and Escherichia shigella bacteria might enter the circulation, and by entering there and reaching up the liver tissue, they could be the ones who would be involved in the pro-inflammatory pathogenetic pathway induction in the liver. But this has to be further analyzed and seen in other publications. So I also wanted to present other works which my group is also doing. These are not the still published data. So we have been working on the circulating microbiome with a gastric cancer. It's the work from my PhD student Daria Nikitina. So she has also this unique cohort of gastric cancer patients and the control individuals, where we also had a look at the circulating microbiome, because gastric cancer is also known to have an increased leaky gut, and that there is a compositional change in the gut microbiome associated with this pathogenesis. And what we saw, that again, the diversity of the microbiome, of the blood microbiome, is decreased in the patients, and we identified differences on the phylogenetic level and identified two bacteria. So it was again Escherichia shigella and another one which was increased in the patient group, and another one was Collinsella, which was decreased in the patient group. So we could detect Collinsella in 30% of the control individuals and only in 3% of the gastric cancer patients. We then did the bacterial network analysis, and what we saw that control individuals have a much denser bacterial network compared to the gastric cancer patients. And when we did the correlation analysis, we, for example, identified that the increase of Escherichia shigella negatively correlated with the decrease of so-called probiotic bacteria like Lactobacillus. And we also further have done the ROC curve analysis, and we saw that we could use Escherichia shigella and Collinsella to distinguish the samples between the gastric cancer patients and the controls. And finally, I wanted to present a very exciting project which is being done with Professor Clovens and Professor Elling Ork and the group from Norway, Professor Johannes Hoff, which aims to distinguish or to identify bacteria which could be used to monitor patients and to monitor their disease activity. And here we are talking about the gut inflammation. So it's ulcerative colitis patients and their comorbidity. So it's primary sclerosing cholangitis, which affects liver. And the study is unique, not only that we would be analyzing the circulating microbiome, but we would be using animal models to analyze how the bacteria could enter the circulation. So we still do not have a lot of results. We are at the starting point where we have been collecting samples from the biobanks. And, but as a very first step, which I have mentioned also with all this contamination and working with these difficult samples, we have done the benchmark analysis, as we knew that if we approach the biobanks, we would be getting very low amounts of a starting material, because usually with the microbiome, circulating microbiome, you would expect to start working with 1 milliliter of the blood or 1 milliliter of the plasma. So here we knew that in the mice we can expect not more than 200 microliters. So we have tested different blood tubes, different starting amounts of plasma, and different extraction kits which were in the market on that time and could ensure us that we would get something out of these small amounts of the starting material. And what we see in the UNIFRAC analysis, that the only factor which affected finally the composition of a microbiome is the kit itself. So the kit which you are working with affected the compositional differences. So you see here at the very left side, right side of the slide, that you can see it starting from the highest phyla level and going down to the genus level. So it's very important to work uniformly and to know which brand you would be using throughout the projects, not in order to get the interpretable data. So thank you once again, and thank you all the partners. And that's my group and Professor Limas Kupčinskas, who might some of you know, who is the founder of our group. So thank you.
Okay, thank you. Thank you for presenting these really very correct and new results on this quite a difficult site of microbiome on the blood microbiome and so on. But yeah, please, maybe somebody has questions.
Yeah, I have. I'm Carsten Christiansen from Copenhagen. Thanks for a nice lecture. I was wondering, do you have any comment on, would you prefer serum or plasma EDTA or for your analysis? We have been trying different, or full blood.
So it's again what question you would be addressing. So
yeah, but
so with
looking for
with
the
whole
microbiome.
Blood, with the whole blood, you would expect that you would get like bacterial cells. And with a plasma and the serum, you would expect that you would get just the DNA pieces which go into the circulation due to the leaky gut as a signaling molecules to other organs, just to maybe induce inflammation or to send a signal. So yeah, it's again, you have to be consistent with what you're working.
Sure, but in your, I mean, you had different types of samples.
No,
so
we just had the tubes, different tubes, but we have taken only the plasma from there, or the serum from there, and we did not see any difference when we compare plasma or serum, which were collected using different tubes, because there were inconsistent data throughout the literature, one saying one thing and the other thing. So throughout our study, we just wanted to check.
Okay, thanks.
Okay, other questions? I may have one question. If you see this increase, for example, on Escherichia or Shigella, and can you correlate it with the gut microbiome, that you see the same tendency if you have, or you maybe are planning to do this?
Yeah. So unfortunately for this liver cirrhosis cohort, we did not have stool samples. So it was one of the drawbacks of the study, but we already knew from the literature, it was extensively studied, that there is an increase of these bacteria, which has led us to make this, to propose this hypothesis that certain bacteria or their components enter the circulation and could contribute to the pro-inflammatory pathogenesis pathway. But in this ulcerative colitis cohort, we will have the patient material sampled, blood plasma, as well as the stool samples for